All right, let's get into it. Today, we're going to be medical detectives. We're going to follow the trail of a single medical sample to see how scientists in the lab track down the invisible culprits, the bacteria that make us sick. It's a real-life CSI, but on a microscopic scale.
I mean, you've probably thought about it, right? A sample gets taken, it vanishes behind a lab door, and a few days later, you get a result. So what on earth happens in between? Well, it turns out a whole fascinating investigation kicks off, and we're about to follow every single clue.
Now, to you and me, this might just look like some cool patterns on a petri dish, but to a microbiologist, this is the crime scene. Every little detail here is a clue. See how the bacteria are interacting with the red blood cells in this dish? That alone tells the scientists a ton about the kind of suspect they're dealing with.
So here's our game plan. We're going to follow 5 key steps in this investigation. First, we'll get to know our suspects. Then, we'll see how we can actually see them. After that, it's all about growing the evidence, making a final ID, and then picking the right weapon to take them down. Let's get started.
Okay, first up, the invisible suspects. Before we can catch our bad guy, we need to know a little bit about what we're looking for. What even is a bacterium?
So here's your basic suspect profile. It's a single cell, super simple. Its DNA isn't neatly packed away in a nucleus like ours, it's just kind of bundled up in an area called the nucleoid. It's got a tough cell wall to protect itself, and some of them, like this guy, have this little tail, a flagellum, that works like a propeller to zip around.
Now, any good detective knows the first step is to narrow down the suspects. And in bacteriology, the first big move is to divide all bacteria into 2 main gangs, the gram-positives and the gram-negatives. The whole difference between them, it all comes down to their cell walls. And trust me, this is incredibly important.
And this shows you exactly what I'm talking about. On the left, that gram-positive cell has this super thick, exposed wall. Think of it like a big, thick chain-link fence. But on the right, the gram-negative cell has a way thinner fence, but it's protected by this extra outer membrane, kind of like a brick wall in front of the fence. This one simple difference, thick, exposed fence versus thin, protected fence, is the key to our entire investigation.
Okay, so we know there are 2 main gangs of suspects. Now it's time to actually get eyes on them. Which brings us to part 2, making the culprit visible. So the first tool out of the bag is the classic light microscope. This thing has been our window into the microbial world for ages, but there's a problem. Looking at live bacteria under a microscope is like trying to spot a clear marble in a glass of water. They're practically invisible, so we have to stain them. And this is the most important stain in all of bacteriology. It's called the Gram stain, and it's a genius 4-step process. First, you hit all the cells with a purple dye. Then, you add iodine, which acts like a lock, trapping the purple dye inside. Now here's the clever part. You wash it with alcohol. That thick fence on the gram-positives, the alcohol can't get through, so they stay purple. But on the gram-negatives, the alcohol dissolves their outer wall and washes all the purple right out. So for the last step, you add a pink stain, which colors the now clear gram-negative cells.
And voila! Here's what you get. It's amazing! Under the microscope, you can instantly tell the 2 gangs apart. The purple guys are gram-positive, and the pink ones are gram-negative. We just got a massive clue. We know which gang our culprit belongs to, and we can even see their shape. Are they little round balls called cocci, or are they rod-shaped bacilli? The case is really starting to heat up.
Okay, on to section 3, growing the evidence. Seeing 1 or 2 bacteria under a microscope is great, but it's not enough to solve the case. To run more tests, we need more evidence, a lot more. We need to grow an entire army of clones from just 1 suspect. So, how do we do that? We give them food. And not just any food. These are different types of culture media, and you can think of them as specialized diets. Some are selective, meaning they have ingredients that only let certain bacteria grow, kind of like a secret password. Others are differential, which means they'll make different bacteria turn different colors, giving us even more clues.
So the key here is to isolate a single suspect. A lab tech will streak the sample across a plate, thinning it out more and more. The whole point is to get individual bacterial cells all by themselves. Then you pop it in an incubator, and each one of those single cells divides millions of times until it forms a visible dot called a colony. That dot? It's a pile of millions of identical clones. We've just captured and cloned our culprit.
And this chart shows just how fast that cloning happens. After a short lag phase where they're just getting settled, the bacteria hit the log phase, and that means exponential growth. One cell becomes 2, 2 becomes 4, 4 becomes 8. It's an explosion. In just a few hours, we have way more than enough evidence for our final identification. And that brings us to Section 4, the final identification. We've got our suspect in custody, we know if it's gram-positive or negative, and we have millions of clones. Now we need a name. And this is where modern tech has completely changed the game.
You know, it wasn't that long ago that identifying bacteria was a long, slow grind. Scientists would have to run dozens of different chemical tests. They'd check to see if the bacteria made certain enzymes by looking for bubbles, like you see here, or if they could eat certain sugars by looking for a color change in a tube. It was a slow process of elimination that could take days.
But today, we have this monster. This is a MALDI-TOF mass spectrometer, and it is a total revolution. It has taken the identification time from days down to minutes. Seriously, it's like going from using a dusty old card catalog to using Google. And that speed makes a huge difference for patients, letting doctors get them on the right antibiotic much, much sooner.
And how it works is just so cool. A tech takes a tiny smear of the colony, and a laser zaps it. This makes the bacteria's proteins fly through a vacuum tube. The heavier proteins are slower, the lighter ones are faster. The machine times this flight pattern, creating a unique signature, basically the bacterium's protein fingerprint. It then instantly matches that fingerprint against a huge database, and bingo, positive ID, case closed on who the culprit is.
But wait, we're not quite done. We've got a name for our bad guy, but that brings us to the final and maybe most important step, choosing the right weapon. Because now we have to answer the question that really matters to the patient: what's going to work against this thing?
This is how we figured that out. The bacteria are spread out all over a plate, like laying down a lawn of grass. Then, these little white paper discs, each soaked in a different antibiotic, are placed on top. And look, where the antibiotic works, it creates a clear zone of inhibition, a dead zone where the bacteria just can't grow. The bigger the zone, the more sensitive the bacteria are to that drug. No zone? That means they're resistant.
And doing this test is more critical than ever, all because of antibiotic resistance. This diagram explains the problem perfectly. In any big group of bacteria, a few might just randomly be resistant to a drug. So when you take an antibiotic, it wipes out all the susceptible ones, but it leaves the resistant tough guys behind. And with no competition, they are free to multiply like crazy, and suddenly you have an infection full of resistant superbugs.
And that leaves us with one last really big question. This whole journey, from sample to result, it's a constant race against the clock. It's a battle of wits between our scientists and these microbes that are evolving at lightning speed. We get better at finding them, they get better at hiding from our drugs. So the real question for the future is, as bacteria keep building new defenses, how will our weapons have to evolve to keep up?